So let's talk non-typical cell walls. So the ones that you will always hear the most often about are going to be the peptidoglycan cell walls. Um, but we have some others. So this is Perowin. Um, yeah, he's a pain, and he's being a pain now. Acid fast bacteria are one of the more unique ones. Mycobacterium species, Nocardia species, they contain waxy mycolic acids instead of peptidoglycan, but they are considered to be modified gram positives because of how they react to gram stains. Archaea, remember, Archaea is going to be our extremophiles. Um, they are prokaryotes, but they are classified in their own domain. Remember, extreme environments, volcano vents, hot springs like in Yellowstone, uh, glaciers. They're not acidic environments or salty environments, even like the salt sea. They do not have peptidoglycan in their cell wall at all, but they still have one. So it's not a typical cell wall. Um, we also have those bacteria that are cell wall deficient, meaning that they have no cell wall at all. Um, the cytoplasmic membrane instead stabilized by sterols. It basically helps the bacteria resist the same osmotic pressure over inflation because of water content. So sterols, you might think of cholesterol because that's actually one of the things that helps our cell membranes stay more stable. But in this case, it's probably a different one. Excuse me. So we had the appendages, sorry, the cell wall. We're coming in even more. Now we're talking about the cytoplasmic membrane. So our cytoplasmic membrane is a phospholipid bilayer. A phospholipid kind of looks like a little monito with two legs. Okay. And at the top, you've got what we call the head, and at the bottom, you have the two tails. So this phospholipid has some unique properties. And if you've worked on the videos for, I think it's chapter 1 and chapter 2, you saw that part of it's water loving, part of it's water hating. And because of that, when they come together, they form a bilayer. Remember, bi means two. So you have, whoops, one side with the heads facing out, and the other side with the heads facing out. The best way I can think to describe this, think of a basketball. I know basketballs are this big, but just go with me on it. Imagine if we could cut a basketball in half and it stayed the same shape, so it was still half a sphere. If I were to touch the inside of that basketball or the outside of that basketball, I would be touching the heads. Okay? The plastic in between those two layers of heads is where these tails are going to be. Okay? So phospholipids make up about 30 to 40% of a cell membrane's mass. And the thing with phospholipids is that they're malleable, they're flexible. So if you kind of think about balloons on a swimming pool surface, if you hit the water in a swimming pool, the balloons will start to move around, right? These move and they're mobile. They're not stationary like a crust, okay? The other 60 to 70% primarily of the membrane is going to be proteins that are floating through this membrane. Okay. They basically help to regulate transport into and out of this cell. So the cell membrane itself is responsible for allowing or not allowing things to move. into and out of the cell. So we say that it is selectively permeable. It decides whether you are green and good to go or you're red and you're not allowed to go. Um, Nutrients usually get to go, certain molecules get to go basically in and out of the cell. And this phospholipid bilayer is kind of like a bouncer to the cell. The tails, remember, do not like water, which means they don't like anything that has charge to it. So if you have charge and you try to get past the tails are going to go, I don't like you. And they're not going to let you actually pass. So if you're something that has charge and you still need to get inside the cell, you actually have to find a protein, because the proteins are the things that create gateways past, sorry, past, this is going to be fun, these phospholipids. Okay. So you've got basically a tube like pipe that is right next to the phospholipids that will allow things to go in or out. Okay. Um, Energy reactions and metabolic activities, um, they contain enzymes for things like cellular respiration, which is basically going to be how we use gases. And usually it's involved in ATP synthesis. Remember, ATP, adenosine triphosphate, is just a fancy way of saying fuel. Okay. Remember, I have no organelles inside of a bacteria. So mitochondria that we consider as, oh yeah, that's essential, that's something that helps us to make our ATP in bulk, they don't have those. So the enzymes, I'm going to erase again, sorry, but I've got to erase a big chunk so I can draw this out. When we talk about the cell itself with all of its gamish inside, enzymes are often going to be inside this membrane, okay? And all of those chemical reactions we're talking about are actually happening from the membrane, not just inside the cell where we could have mitochondria, the ATP factories that we have in eukaryotic cells. Okay, so inside of the cytoplasmic membrane, um, the cytoplasmic membrane is completely surrounding all of the cytoplasm. Now remember, cytoplasm is that jelly-like solution inside of the membrane. So if I were to fill a small Ziploc bag with jelly, the Ziploc bag would be the membrane, and then the jelly would be the cytoplasm. So cytoplasm is about 70 to 80% water as far as the mixture goes. But other than that, things that you have inside of that jello, okay, that jelly, are going to be soluble, dissolvable proteins, salts. You know that if you put salt in water, it'll dissolve. Carbohydrates, if I put sugar in water, it'll dissolve. Um, that cytoplasm is the site for nearly all of the chemical reactions that are going to happen inside the cell. Now remember, we have those enzymes that are still located on the membrane, so those are still happening as well. But this big, I guess I could say like room inside of my Ziploc, the jelly with all this stuff in it, is where most of the chemical reactions are going to happen. So inside of the cytoplasm, as I said, most of the action is taking place in there. But you've got kind of a few important structures that are there. The first one we're going to talk about is the nucleoid. Remember, not nucleus. That is completely different. Okay? There is an area. It's not enclosed. There is an area where the bacterial chromosome, the DNA, is found. It can also contain those little rings of DNA that I talked about before, the little plasmids. Um, Excuse me. Plasmids are not something that is required. It's kind of an optional little piece of DNA that can give the bacteria extra traits that it didn't have before. The thing about having this little plasmid is that it can have genes, let's say bacterial protection genes. Okay? It can have these extras. Maybe it gives them the ability to use maltose as a fuel source, whereas before it didn't have the ability to do that. So you've got, I'm sorry, I have cat fur on my face. You've got an area where DNA is found, and that includes any extra little rings of DNA that are plasmids. Okay. And again, plasmids, they may carry junk, but sometimes they can carry beneficial traits. Okay. Ribosomes. Ribosomes to me have always looked like hamburger buns. You have the top bun, you have the bottom bun. Okay. Ribosomes are responsible for protein synthesis. Synthesis is a very fancy way of saying to make something. So these two units, the large subunit at 50S and the small subunit at 30S, S is just size, okay? They are responsible for making proteins inside of our bacteria. So you've got 50 and 30S combined to make a total size of 70S. Yes, I know the math doesn't work. I'm not the one who invented it. Sorry. The reason that the number is assigned, though, is because we want to make the distinction between ribosomes in bacteria versus ribosomes in eukaryotes. Ribosomes in bacteria are definitely smaller because when you look at ribosomes in eukaryotes, they are 80S. Okay? Yes, they look the same. Yes, they've got a large and a small hamburger bun. That all is accurate. But push comes to shove, the ribosomes you find that are responsible for protein synthesis in bacteria are smaller than those you find in eukaryotic cells. All right. So the third structure we're going to talk about are the inclusion bodies or the granules. These are basically storage closets, okay? If they have extra nutrients, they can put them in this little bubble. If they've got extra gas, they can put it in this little bubble. Any other molecules that they will use, they can store in an inclusion. Again, it's a storage closet. You can call them granules because they look like granules uh, in a microscope, but um, they're basically just a place to put things that we will find useful. I can't use them now, but I can use them later, kind of thing. So let's talk endospores. It's a survival pod. Okay. Endospores are dormant bodies formed by certain species of bacteria. Not all bacteria can make endospores, um, but those that can actually do have quite a bit of an evolutionary advantage. Um, what causes them to form? Hostile conditions. If a bacteria goes from being in a nice, warm, moist environment with plenty of food to all of a sudden an arid, like hot, dry environment and there's no food in sight, it will switch gears and go into endospore formation. Okay. Um, Basically, just like survival pod leans towards stating, it helps them to survive until favorable conditions come back around. So I'm sure you guys know that toads that we see out here in South Texas only really come around after rain. After rain, you will find them on your porch, you will find them in the garden, you'll find them in your yard, but before that they're not really present. And one of the reasons is because when it starts getting dry and arid and everything else, they'll actually go into a state of hibernation and wait until it rains again and they can come back around and start doing what they do. So, the species that are most closely associated with having endospores are Bacillus and Clostridium. Okay? These are the ones that you hear about making endospores the most often. They are not a reproductive body. Okay? Literally, it's the same singular bacteria going into hibernation. It's not like a bear that goes into hibernation to make more bears. If they're hibernating, it's the same bear when it wakes up. Okay? So, when we are talking about making spores, the formation, of spores, we call that sporulation. That is just the proper way of saying it. It's the proper term. So, when you go from being a spore to being active, we say that it's the return of the spore to the vegetative state, meaning it is a normal bacteria. Um, we say it's the vegetative state germination. I'm basically back in favorable conditions, so I'm going to become active again because I've got food, I've got water, I've got the perfect conditions to do this. And with endospores, that switch from being hibernating to being active can happen real fast. Okay? So, why do we care about endospores? Because medically, they are really important because they can mess up an entire hospital. Okay? They are very resistant to heat. So if you think, putting something in boiling water is going to help with endospores around, it really isn't. They will survive that. Okay? They are very resistant to soaps and disinfectants. So if you think I'm just going to wipe down the counter with some soap and water, it'll be fine. Not if we're talking about endospores. They're in a hard shell that's protecting them. They're fine with soaking in soap and water, whatever. They don't care. This is a scary one for us, this last bullet here. In a hospital setting, endospores can be very hard to control because the way that you get rid of them may not work for the environment that you're in. So it's a balancing act, honestly. So if you look on the right hand side, this is a spore. Inside, you will notice that there is this little bubble. This is actually the endospore. This is the thing that will rise again. This whole outer covering, the sporangium, is the dormant part. It's that hard shell that's protecting it. So I'll move myself down here. All right. Typical sporulation. Okay. We've got a vegetative cell, of course. Where's the button? Okay. We've got a vegetative cell. Remember, vegetative just means that it is active, it's doing what it does. Okay. We've got a vegetative cell that starts getting depleted of nutrients. In other words, its environment is becoming less and less favorable. It's not working as well as it did before. So, when I kind of get kicked into sporulation gear, the first thing I'm going to do is I'm going to duplicate my genetic material and I'm going to separate it. Basically, I'm making a copy and I'm isolating those two copies. The cell will septate. It'll form a septum, okay? See that division point right there? Okay? Into a sporangium and a forespore. Okay? So, the forespore is this little dude here. This over here is going to be the sporangium. All right? So, the sporangium, the bigger half, okay, is going to engulf the forespore. It's basically going to surround it, okay? And that's going to allow the next steps to occur, further development to occur. So the sporangium begins to actively synthesize the layers of the spore around, okay, the forespore. So I'm making layers of protection, okay? I'm going to move this out of the way. All right. And this is going to switch again, I think. Okay, yeah. Okay, so sorry. The cortex, which is outer layer, and the outer coat layers are starting to get deposited. So this is that copy of the genetic material inside of these layers. Think of a peanut M&M, okay? And my peanut is going to be on top of my genetic material, and then you've got the chocolate, and then you've got the candy. So there are layers basically surrounding this little bundle of genetic material. If you look, this guy is kind of becoming smaller. The genetic material in the outer portion, by the seventh step, it's starting to fall apart. Literally, it's being cut into pieces. So I still have my little protected endospore that is now mature. When this outer portion dies away, okay, I've got the free spore that has the core, the DNA, the cortex, a spore coat, an exosporium, layer upon layer of protection, protecting this genetic material. Um, if I don't hit a favorable environment, I stay at step 8. If I'm not in a favorable environment, I just stay in hibernation and I don't do anything. But if I hit a favorable environment, I will go through the ninth step, germination. The spore will start to swell and it'll release that hibernating vegetative cell, and then we've got a cell that can go on and infect. In a hospital setting, um, these spores can sit in equipment for months, for years, and they never reactivate. But then you take that device and you use it on a patient, and all of a sudden it's in the perfect environment. It germinates and it creates active cells that can now infect. This process is a nightmare for a hospital because this process, especially if you've got spores in your space, can cause so many issues for patients and doctors and staff and everybody in a hospital. For patients, it can literally be deadly. If you're transferring spores into their body and those spores go active and are infectious, they're pathogenic, then it can kill the patient, especially if they've just had surgery or something like that and their body is already dealing with damage. To add infection to that is just like adding insult to injury. All right. So that's the typical sporulation cycle. Now, Archaea. We kind of already touched on Archaea a few times now, saying that they are a prokaryotic cell, but they live in very extreme environments. So bacteria are not the only prokaryotes that we have. The Archaea are considered a different domain than bacteria, um, even though both are physically kind of the same. So, what's the difference? Why do we separate them? Both have a cell wall. That is true. But remember that peptidoglycan that we mentioned was so important before? Archaea doesn't have it. Okay? When you compare Archaea to eukaryotic cells and Archaea to prokaryotic cells, they actually have more characteristics in common with eukaryotic cells than they do with bacteria. Um, Ribosomal RNA sequences, protein synthesis, ribosomal subunits. Remember, I said they're different sizes. The bacteria have smaller than the eukaryotes do. Yeah, these guys have the size of ribosomes as the eukaryotes, which isn't English. These guys have the same size ribosomes as eukaryotes. So that's a big whoa, wait a minute. That they're not the same. So we definitely needed to put them in their own unique category or domain. Something else. Archaea are the extremophiles. They are not found, in dirty dishes. They're not found in an infected wound. They're found at extremely hot or cold temperatures, extremely acidic pH, or extremely basic pH. Um, Very salty environments where if I were to put regular bacteria, they would die. So, one of the reasons that canned goods have such high salt content is because normal bacteria that would cause food to go bad don't like that. So, if you make it extremely salty, the chances of that thing growing in there are very low. Notice I didn't say it'll never happen, I just said it's very low. With the extremophiles, though, the Great Salt Sea, the one that, there are no living fish in because it's so salty it would kill them, you can find Archaea there. The hot springs in Yellowstone that if, a buffalo falls in it, it literally boils the flesh off of their bones. You will find them there. There are other hot springs that are so acidic, it would literally burn anything that fell into it within a few seconds. Excuse me, except in this case, acidically, not temperature wise. Again, you will find them there. This is key with the archaea, though. If they are comfortable living in that environment, when you change it, if you try to take them out of there and put them onto normal growth media at a normal pH or normal temperature, they're going to die because their perfect environment to live in is in that hot spring or in that acidic spring or in the Great Salt Sea or in a volcano vent or in an iceberg or whatever. No matter what, they're used to being in that place. Be like me saying, okay, I'm just going to put you on the surface of the sun. You wouldn't do very well either. But if there was a volcano dragon monster living on the sun and we moved them to Earth, it would be like putting them in ice water. It just wouldn't work out real well for them. Okay, so terminology for the classification of prokaryotes. Bacteria are classified just like other organisms. You've got the kingdom, phylum, order, same thing, kingdom down to species. But they also have divisions and classifications below the species level. We can call them the subspecies, the strain, or the type. These are terms used to designate bacteria belonging to the same species that have different characteristics, whether that's physical characteristics, chemical characteristics, or biological characteristics. Having a great time with English today. Serotype is also a term used to give members of the same species that stimulate different patterns of immune response in a host. So, Vibrio cholera, for example, is the agent that causes cholera worldwide, but it does have multiple serotypes. Okay. The two critical ones, the ones that are the most well known, okay, make these terrible toxins. Okay. They are designated as Vibrio cholera 01 and Vibrio cholera 0139. These are the two most deadly forms of cholera that create toxins that affect the human body. All right. So, focusing on a disease, tetanus, commonly called lockjaw, is caused by the bacterium Clostridium tetani. Okay. I'm sure you've had your tetanus shot. It's to prevent this. So, lockjaw was an awful disease. The thing about Clostridium tetani is it can be found everywhere. It's in the soil, it's on the concrete, it's in nails, it's everywhere. So what would happen was somebody would get cut on something and soil would get into it. They would get cut on an iron nail, whatever. And this particular bacteria actually started to produce a toxin. Okay. Excuse me. Titanospasmin. Okay. The other thing that's dangerous with Clostridium tetani is forms endospores. So it helps it to survive in the environment that we live in, whether that is on ice when it snows, or in the soil, or on the concrete when it rains. The endospores are there and they're perfectly happy being there, just in their little state of hibernation. The agent itself does not need oxygen, so it grows really well in puncture wounds. So when you puncture through your skin, it's not like there's air readily hitting it, especially depending on how deep it is. Okay. So in a puncture wound, they germinate. The spores come to life and they start multiplying. Again, the danger is that once it's germinated and it's active, it starts to produce this toxin, titanospasmin. Okay. It produces paralytic symptoms. Basically, it causes your muscles to contract. The reason they called it lockjaw is because these people's jaws would just completely lock. There's a very famous drawing. If you look it up on, excuse me, if you look it up on Google, look up lockjaw. It's a hand drawn picture, and the patient is on his back, but he's literally bowed over the bed. His toes are even curled, his hands are curled, and he's got this grimace on his face. Back in the day, if you got lockjaw, there was nothing they could do. They would usually pop out one of your teeth and feed you through a straw until you died. Once that toxin got to your diaphragm, it froze your diaphragm and you would suffocate to death. There was nothing they could do. They had no way to treat it. So, what was invented? A vaccine. I know we all know what a vaccine is in today's day and age, right? So, when you get your tetanus vaccine, what they are doing is they are giving you parts and pieces that your body will recognize and be able to create an army against. Okay? So I've got these little parts and pieces. I don't have the full, I didn't give you Clostridium tetani. I just gave you parts and pieces of it so that your immune system would start creating an army against those pieces. It would be like having the fingerprints of a criminal, having the ear print, because the ear print is actually just as unique as a fingerprint. Um, so I've got the ear prints, I've got fingerprints, I've got toe prints. I've got a way to recognize the, I don't know, serial killer. Okay. Did I send the serial killer to your house? No, but I gave you ways to recognize him. Even if I give you a picture of the serial killer, that would be a way to recognize it. When you've got vaccines, they're giving you bits and pieces for your body to react to create an army against. Okay. That means that if it ever does get into your body, your immune system already has an army there to deal with it. So you don't get lockjaw. Instead, it comes into your body and your body can kill it relatively quickly. Instead of it taking 7 to 14 days, and by the time that happens, this isn't going to work anymore. Okay? This is preemptive. I am preparing your body before you get sick. If I were to wait until you were sick to give you this, it's still going to take 7 to 14 days. And by that point, this is already too far gone. Okay? So, it causes four types of tetanus, four types that are known. Okay? Generalized is the most common. It's the picture from Google. 80% of the cases are this. Now, until more recently, most people would go and get their tetanus vaccine. Um, Nowadays, they see people coming in with tetanus because they didn't get vaccinated against it. And again, it's one of those things where you can kind of watch. Some of the muscle spasms that are created because of tetanospasmin are actually strong enough to break your own bones, just to kind of give you an idea of how bad this disease is. Um, Very rare forms include neonatal, local, it's only one part of the body, and cephalic, which means it affects your head. Okay. So, just to give you an idea, this is one of the diseases that it has a bacterial cause that can be deadly. And because it was as deadly as it was, there was actually a vaccine created so that, God forbid, you get exposed, you actually have a way to fight it. Okay, so that is the end of, sorry, chapter 3.